Good afternoon, guys. In today's lab, you will need the following: one russet potato, one blender, two coffee filters, one 10 ml and one 100 ml graduated cylinder, four 50 milliliter beakers, a bottle of hydrogen peroxide, 3%, a standard hole punch, a large piece of filter paper, some DI water or distilled water, a 500 milliliter flask, and a 40 milliliter reaction container. We're actually going to be using other things as well, so please watch carefully. What I'm going to first do is I'm struggling sort of to cut a potato. As you can see, I couldn't find a proper knife, so I'm using the sharp blade of a spatula. We're cutting the potato into chunks so that we can blend it and puree it in the blender. So here I go, and we're going to take a break here, and we're going to cut to blending. Alright, but before we actually blend the potato, we actually want to fill up a 100 milliliter graduated cylinder with 100 milliliters of water. We're actually going to add 200 milliliters of water to the potato in the blender. There it is, the 100 milliliter graduated cylinder, adding the water. Alright, we'll do it one more time. We got to fill it up. So we're going to wait and watch me perfectly fill up 100 milliliters of graduated cylinder. There it is. And we pour. Okay, now we're going to add the top, and we are going to puree, that is to say liquefy, the potato with the water in it, of course. The idea here is that we are blending the potato to get rid of all of the other cell products, and we're trying to isolate catalase, that enzyme that we're going to be looking at today. We're going to be looking at catalase, and in the liquid will be the enzyme, and we're going to try to filter out the chunks of potato from the puree. So we're going to take, to do so, we're going to take a 500 milliliter flask, as well as a funnel, plus 2 pieces of coffee filter. The idea is that the coffee filter is going to catch the potato and let the liquid flow through it. Okay, so we're going to add that puree, and in a second we're going to see a close-up of the liquid filtering through. So here we go. It's sort of difficult to see. Also, the image is sort of distorted because the phone that I was using was turned upright, and so the image has been sort of stretched out a little bit. But what you can see coming out of the funnel is the liquid from the puree. The idea here is that we are collecting the liquid from the potato to isolate the catalyst. That is to say, the enzyme catalase. Alright, and in order to use this catalase mixture, what we're going to do is we're going to pour it from the flask into a beaker, so it's more easily accessible. Now what we're observing is the actual basic procedure to measure enzymatic activity. And in fact, the first part of the procedure asks us to test the effect of enzyme concentration on enzymatic activity. So what I'm doing is I'm filling 4 50 milliliter beakers with different concentrations of catalase. In that first beaker to our right, we see that I have added, using distilled water and a graduated cylinder, 10 milliliters of water alone, no catalase. So that's a 0% solution. The next beaker from the right, I've added 7 milliliters of water, and now I'm measuring out 3 milliliters of catalase. In the third beaker from the right, I will add, I've added 3 milliliters water, and then I'll add 7 milliliters of catalase. And in the last, or in the leftmost beaker, I will add a complete 10 milliliters of catalase and no water. So as we're moving from right to left, we're going from very dilute solution to a very concentrated solution. As you can see, I'm measuring out a very precise measurement of catalase solution. I believe I'm measuring 7 milliliters into beaker 3. Alright, again, take a look that I'm using the pipette to measure out that catalase solution, also using the graduated cylinder. Alright, I'm measuring out 10 milliliters of catalase solution to add to the leftmost beaker. And that should be it. There we go. Okay. Keep in mind now, I'd like to homogenize those solutions, making sure everything is mixed well, especially those middle ones, because those middle ones are catalase and water. Now I'm measuring, or at least just observing, to see that everything should be about 10 milliliters. Right? Everything should be about the same volume of solution. That's an important part to control our experiment here. So now I'm setting those little beakers off to the side. And this is where our procedure, this is where what you will do in our procedure. You will prepare the beakers, then you will use what I just showed you, the 40 milliliter reaction container, the hydrogen peroxide, and again, our 100 milliliter graduated cylinder. It's also smart to use a flask to measure out, we're going to measure out 40 milliliters of hydrogen peroxide. So we're going to use the flask, not the flask, the funnel. We're going to use the funnel and the graduated cylinder to measure out 40 milliliters of hydrogen peroxide. Once we've got 40 milliliters, we can then take off the funnel, set it to the side so we don't knock it over, then pour it into the reaction container. This reaction container is the container that we will be looking to see the reaction. Now what I'd like you to do is take out a hole punch and the filter paper, and we're punching out little holes of filter paper to control for the size, right? Because we want to make sure that our pieces of filter paper are of equal size. Take your forceps. All right, I'm talking to somebody. Take your forceps, dip them into a catalase solution, hold it there for about 3 seconds, tap it off, hold it there for another 3 seconds, tap it off again. Now you will need to push that hole punch to the very bottom of the reaction container. And now, as soon as that touches the bottom, what you will do is you will start timing. And you will be waiting and timing to see how long it takes for that hole punch to lift and float all the way to the top. It will float because of the decomposition of the hydrogen peroxide due to the enzyme catalase. I'm punching out a couple more holes, and we're going to see it again. Okay, using the forceps, we're going to make sure you hold on to that filter paper nice and tight. Dip it into the catalase solution. And then again, place it down to the bottom of the reaction container and start timing. Again, we're measuring the time it takes the enzyme to decompose hydrogen peroxide, creating oxygen gas to lift or float that little hole punch. Once it hits the top.